Chapter 1 is over the scientific method, and the scientific method is composed of 5 different steps, and this is the process you go through whenever you are going to be designing an experiment, testing an observation, all that. This is the process, the method that scientists use to make discoveries and draw conclusions about the world around them. So the first step is you make an observation. So you see something, you see lightning, and you wonder what causes lightning. You're observing the natural world around you and trying to see what is something new and how that process may work. The second step is you propose a hypothesis explaining your observation. So you might say, I see that lightning occurs more during thunderstorms, and so I think there's a Greek god of thunder that throws lightning bolts, and that's where they come from. That's actually kind of a bad example of a hypothesis, because remember, a hypothesis has to be testable, and you have to be able to prove it false. So if you can't prove it false, if it's not falsifiable, and if it's not testable using measurable data, measurable results, then it's not a good hypothesis. So you cannot use supernatural explanations for a good hypothesis. The third step in the scientific method is you're going to make a prediction about the results of testing your hypothesis. So after you come up with your hypothesis explaining your observation, you're then going to predict what you think is going to happen whenever you design a test and test your hypothesis. The next step is the actual experiment, designing the experiment to test your hypothesis. A good experiment is going to limit bias as much as possible. Remember that bias is the attitude of the experimenters or the people participating in the experiment and how that's going to affect how they interpret the results. So you don't want bias. Bias is a bad thing. You want to limit that as much as you can because you want to just look at the data and see what the data shows. So in your experimental design, to limit bias, the gold standard, especially for medical research, is a double-blind, placebo-controlled, randomized experiment. So what that means, the double-blind part means that the people that are assigned to the different groups, the different testing groups, don't know which group they're assigned to, so that's the single blind. And the double-blind part has to do with the people, the researchers, who are analyzing the results and gathering that data. They don't know who's in which group either. So no one that's actually involved in reporting data or gathering that data, compiling it, nobody knows who's in the test group and who's in the control group or who's gotten the drug and who's gotten the placebo. So that's the next step, is that placebo-controlled. So a placebo is like a sugar pill. Basically, it's something so that you're kind of fooling the person taking it into not knowing whether or not they're actually taking a drug or not. Because there's always going to be a placebo effect where somebody takes a medication and thinks that it helps them, even though it may not have done anything at all. There is kind of a psychological effect to medications. So the placebo effect and the placebo-controlled experiments kind of factor that in, that there is going to be a psychological effect of the medication. And so you're trying to measure the differences between the groups once you take away that psychological effect. And then the last part of that is a randomized experiment. And what that means is that the people that are assigned to the different groups, they're assigned randomly. The experimenter's not saying, well, I want all the males in this group and all the females in this group, or I want all the young 20-somethings in this group and all the old ones in this group. It's completely random because you're trying to get a good sample size in your results. If you cannot do a double-blind, placebo-controlled, randomized experiment because it's not ethical, so let's say it's a very iffy test drug and they don't know if it's going to hurt or hinder someone, then what you can do is you can do a correlational study, and there are different types of correlational studies which your textbook goes over, and you do need to be able to match those up, the different names with what they are. Those correlational studies can be used to look at old results and kind of see if there's a correlation between two variables, between two factors. But as we'll discuss down here under Analyze Results, correlation does not equal causation. Just because there's a correlation doesn't mean that one thing is causing another. And that's a very common mistake that lay people would make when they're looking at scientific data. Alright, so in your experiment, there are 3 types of variables. There is an independent variable, there is a dependent variable, and then there are the controlled variables. So the independent variable is the variable that the scientist is tweaking. So this is the one that you control. So you as the scientist control the independent variable. The dependent variable is what changes as a result of what you've tweaked. So in your study guide, there's an example about an experiment that's designed to see how well a plant is going to grow under certain conditions. So the conditions that you change, so if you change the type of soil, if you change the amount of water that plant is getting, whatever you change, that would be the independent variable. And then the dependent variable is what you're measuring as a result of that. So if we're talking about how well a plant is growing, you might measure the height of the plant. So the independent variable would be the type of soil that you chose, and then the dependent variable would be how tall the plant grows. The important thing about these independent variables and dependent variables is to remember that you should only have one independent variable in each experiment, because you're trying to show a link between the independent variable and the dependent variable. So if you've got more than one thing that you've changed between the groups, then you won't know what's causing what. So in a good experiment, you've got one independent variable, and you're measuring a dependent variable that's easy to measure. So it has numbers, it has data that you can gather to see, an exact height on the plant, or how many leaves the plant has, or how long the leaves are, or how many fruit the plant produces. So you want definite numbers that you can record to analyze this later. And then the control variables are everything else in the experiment that you're keeping the same. So you're keeping it the same between your test subject group and your control group. The control group, if we're talking about a medical experiment, will be the group that receives the placebo, whereas the test group, the test subjects, would receive the actual medication. So you want all those other variables to be the same as much as possible. So if we're talking about a medical experiment, then ideally you would want identical twins, kind of the perfect situation there where one got the drug and one didn't, and how did they react to that drug? But of course that's very hard to do. So what you do is you get a big sample size, so that even though they're not going to be exactly the same, they're going to be some older people, some younger people, some males, some females. If you have a big enough sample size, all of those factors will kind of even out as you're looking at your results. So you want to have as many control variables as you can, and if you can't control everything, then you want a big sample size so that the individual variations don't matter as much. So step 5 of the scientific method is to analyze your results and then draw conclusions based off of those results. We typically use statistics to help us analyze our results, and there is a section in Chapter 1 that talks about some of these statistics, the mean as far as the average, standard deviation, things like that. Make sure you understand those terms and what they mean as far as applies to the data. So for example, when we're talking about the term statistically significant, that's very important. What that means is that it's unlikely that your results are just due to chance, just kind of the randomness of how the world works. It's unlikely. So if it's statistically significant, that is a good thing. That means the probability of the results being due to chance alone is 5% or less. Now, notice that it's not 0%. You can never assume that there is no chance affecting your results. So if we can get 5% or less being due to chance, then we're happy with that. That makes it statistically significant. So that's what that term means. As we've discussed before, correlation does not equal causation. So an example of this is my mom is recently kind of getting into the paleo diet, which if you aren't familiar with that, it's kind of a grain-free, high-protein diet. So she's been listening to a lot of nutritionists talking about the paleo diet, and they're trying to link certain things. So they're trying to link, let's say, people that eat a lot of fried food or corn or something along those lines, and then their poor health later on, or people that eat a lot of fat and they have poor health or something like that. And so you have to remember there's a lot of confounding variables in those sorts of analysis. So, one of the things the paleo people really promote is a lot of red meat, a lot of meat. And traditionally, a lot of people have said that you don't want to eat meat. That can actually be bad for you if you eat a lot of it. And so you've got to keep in mind that you're talking about the types of people that will be eating red meat, if they're eating it excessively, might have other issues, other health issues that be confounding those results. So they might be the people that tend to exercise less, and be overweight. They might be people that eat more fried foods. So there's a lot of factors involved, and it's hard to tease that apart, especially nutrition, about whether or not one thing or two things really will cause you to be healthier or cause you to be less healthy. Same thing, you'll see people say that such and such causes cancer. Well, that's almost impossible to prove in most cases because there's so many other confounding variables that are involved in that. So correlation does not equal causation. You can say that there's a strong correlation between two things, like a strong correlation between high cholesterol values and heart attacks. But you cannot say that high cholesterol causes heart attacks unless you eliminated every other variable in that study. You can't really say that it causes it. So correlation is not equal causation. And you'll see that a lot in medical studies, that term correlation. So just realize that doesn't mean causation. That doesn't mean that they've proven that one thing causes another. It just means they think there's a strong correlation between them. And more studies probably need to be done to kind of tease apart the other things. Sample size matters. Sample size is very important. If you do a study and you study the results of a medication on 10 people, that's probably not going to compare very well to if 1,000 people take the medication and those results. So sample size is very important. You want a large sample size because that lets you get rid of some of those random little details that can affect how one person metabolizes a drug versus another person. So when we're talking about sample size, another term you should understand is the confidence interval. And so the confidence interval helps you know kind of whether or not you have a statistically significant result. So the confidence interval has to do with the sample size. So if you have a big sample size, you typically have a smaller confidence interval, and that's actually good. So you want a small confidence interval because what that means is that the sample size you picked was big enough that probably your results are not due to chance alone. So this is going to sound weird, it's kind of almost counterintuitive, but you want a smaller confidence interval, not a larger one. A larger one is actually bad because that means that you've got a lot of chance differences in your sample group. You probably have a smaller sample group, and that's not as good of an experiment. Alright, so that's the review over Chapter 1. Make sure you go through the study guide that's online. There are some other things in there that I point out as far as being important and that will be on the exam for this chapter.